everybody. Today we'll be talking about rabies. My name is Ashley.
I'm Julia.
I'm Celeste.
I'm Araceli.
And my name's Zach.
All right, let's get into it. So rabies, it's popularly coined the world's deadliest disease, and rightfully named so, as among one of the only diseases to be nearly 100% fatal upon symptom appearance. Rabies has become one of the most famous and terrifying diseases on Earth. So what is it? Rabies has had multiple names throughout history. The ancient Greek called it Lyssa, meaning frenzy, for the madness that it induces in its host, which actually has developed into its current scientific name, Lyssa virus. Some cultures call it mad dog disease, and some call it hydrophobia, a name after its most famous symptom. But in pedantic terms, rabies is a negative sense, non-segmented ssRNA virus, and we will be talking about all this later. It is a zoonotic infection of the nervous system that can infect almost any mammal and kills by causing encephalitis in its host. And it is transmitted through direct contact with saliva or nervous system tissue from the infected, which popularly translates to a bite. All right. So the first written record of rabies actually appears way back when in the ancient world, nearly 4,000 years ago in the Mosaic Esmuna, which was a law stating that Babylonians must pay a fine if their dog transmits rabies to another person. However, the way that rabies was transmitted stayed kind of a mystery until about 2,000 years later, when a Roman philosopher named Celsus guessed that it was spread by animal saliva. Now, interestingly enough, rabies might actually be the source of one of our most popular mythic creatures, vampires. And there's a couple reasons why people think this. The fact that vampires are biters is pretty incriminating. But also, rabies victims can start acting in really primal, animalistic ways, which could be connected to vampiric transformation. I think the most interesting point of connection between the two is actually vampiric aversions. So rabies can actually make people very adverse to certain things, like strong smells, which could explain why vampires don't like garlic. And this is the kicker, to their own reflection, which could explain the reasoning behind the myth that vampires can't look in mirrors. Continuing on, in the 19th century, Louis Pasteur makes a vaccine, which leads to now. Rabies rates keep trending downwards, and what was once a terrifying and overwhelming disease is now kind of manageable. Okay, so how did Louis Pasteur make this miracle vaccine? The story starts with a boy. The patient in question was Joseph Meister. He was a 9-year-old boy who was bit 14 times by a mad dog. Now, at the time, Louis Pasteur had been experimenting with a vaccine made from the spinal cords of rabid rabbits. He discovered that if you left the tissue to dry, the virulence would decrease. So he used that to make an attenuated strain that he could use as a vaccine. But up until Joseph Meister, he had only tested it on some dogs. So Joseph Meister became his first human patient to ever receive the treatment. Over the next 11 days, Meister received 13 inoculations of attenuated rabies, each progressively more virulent than the last. And miraculously, Meister never developed rabies, making him not only the first human to participate in this treatment, but the first ever human being to be vaccinated for rabies.
Now for the etiology. Rabies infection is caused by the rabies virus, which is a bullet-shaped RNA virus in the family Rhabdoviridae and genus Lyssavirus. Rabies is spread through the saliva of an infected mammal, and transmission is usually through the bite of an infected mammal, but it can also occur when infected saliva gets into an open wound or mucous membranes, like the mouth or eyes. Infection can also occur through the inhalation of the virus in an aerosolized form, ingestion, and rarely through organ transplants. Animals that spread rabies can be both wild and domestic, and most commonly consist of the animals pictured here. 99% of rabies cases worldwide are transmitted by domestic dogs, but 7 out of 10 of rabies deaths in the US were transmitted by bats. Rabies evades the immune system by sequestration in the nervous system. There are 5 general stages of rabies, which are incubation, prodrome, acute neurologic period, coma, and death. Incubation occurs when rabies enters the peripheral nerves and typically lasts 30 to 90 days long. The prodromal stage lasts 2 to 10 days and begins when bare nucleocapsids enter the motor and sensory axons. During the stage, nonspecific symptoms like fever, fatigue, sore throat, and cough occur. Prophylactic therapy becomes ineffective and the mortality rate is almost 100%. Rabies travels along the axons to enter the spinal ganglion, spreads rapidly into the central nervous system, and goes to the periphery and salivary glands, where it can be transmitted to others. Acute neurologic period occurs when the virus has entered the central nervous system. Rabies can be classified as furious rabies or dumb rabies, depending on the symptoms. Furious rabies is when hyperactivity predominates, such as hydrophobia, and dumb rabies is when paralysis predominates. A coma occurs soon after this, which will then lead to death. The vast majority of rabies therapies, once a patient is infected, are palliative instead of aggressive due to the likely outcome of death. Palliative therapies aim to prevent and treat neurological complications due to rabies using sedatives, narcotic analgesics, anti-epileptic medications, and neuromuscular blockers. Under rare circumstances, such as an early stage of rabies, an aggressive therapeutic approach may be used, but in the very low chance the patient survives, they will be left with permanent neurological disabilities. Some treatments for patients with rabies under current consideration consist of ribavirin, alpha, and the Milwaukee protocol. Ribavirin is an antiviral agent that has in vitro activity against rabies infection by inducing mutations such as mismatches and substitutions in viral genomes. Alpha, which we learned about in class, is an immunoregulatory protein in the innate immune system that generates an intracellular environment that restricts viral replication. Milwaukee protocol is when the patient is put into a chemically induced coma to give the brain a rest and allow the immune system to catch up. Once in the coma, the patient is given antivirals such as ketamine and amantadine. This has a current survival rate of around 14%. Since there is no proven effective therapy for rabies, most rabies therapies are centered around prevention. Post-exposure prophylaxis, or PEP, is used for bite and non-bite rabies exposures. It protects the person from developing rabies and prevents transmission. PEP consists of immediate wound cleaning, human rabies immune globulin, or HRIG, and the rabies vaccine, which is given on days 0, 3, 7, and 14. Those who have been vaccinated for rabies just received the vaccine, but those who are unvaccinated received HRIG and the vaccine. HRIG works by neutralizing the virus before invasion of the immune system, and the rabies vaccine is inactivated, so it does not trigger cytotoxic T cell response, but it does trigger the immune system to make antibodies. Pre-exposure prophylaxis, or PrEP, is a series of vaccine doses given prior to rabies contact for people who are in high-risk exposure groups. People who receive PrEP from highest to lowest exposure risk consist of those who work in a laboratory with the live rabies virus, those who have frequent contact with bats, and those who have frequent contact with other rabies carriers, like veterinarians, animal control officers, and wildlife biologists.
The first step of the viral cycle is attachment, and for rabies specifically, the RABV-G protein is what binds to the neural cells. Some of the receptors that the RABV-G protein binds to include the nicotinic acetylcholine receptor, the neural cell adhesion molecule CD56, and the low affinity nerve growth factor receptor P75. And then based on research, it has shown that greater expression of CD56 and P75 receptors equates to greater susceptibility to the RABV protein binding. And then other binding places include the neural cell surfaces, negatively charged lipids, and then the attachment of the RABV-G protein is highly influenced by the pH levels within the environment. And so lower pH ranges, which are 6.5 to 5.6, is the ideal range for the RABV-G protein to actually bind to the host cell receptors. And the second step to the viral cycle is entry, and the virus enters the host cell after attachment through clathrin-dependent endocytosis following attachment. And then this includes 2 entry stages, the early stage and the late stage. So after the virus has entered through means of endocytosis, it will enter into a coated vesicle. The early stage of endocytosis, the vesicle will lose the Catherine coat, so it's being uncoated, and then in the late endocytosis, all the contents inside the vesicle will then be degraded. And after this, the pH levels will lower in the cell to approximately 6.5 pH, where fusion of the viral envelope will occur. And from here, the viral components will then be released into the cytoplasm.
Now onto transcription. It was briefly shown the genome makeup of rabies virus. However, I will go into more detail here. It consists of 5 genes, that being first the nucleoprotein, which is denoted by N, the P protein, which is phosphoprotein, the M protein, which is the matrix protein, the G, which is the glycoprotein, and L, which is large protein that also acts as the RDRP. Once the virus enters the cytosol via fusion out of the endosome, the matrix protein is degraded, and this is because the matrix protein actually makes it so transcription cannot occur. So once it's degraded, then transcription starts. Since rabies is a negative sense RNA virus, that means that it packages its own RDRP, and this RDRP is actually completely competent, which means it can do more than just transcription. We'll get into that later. There are these intergenic sequences between each of the cistrons. Each mRNA contains 1 cistron, and that is for each protein. The intergenic sequences, as we have learned in class, contain slippery U sequences that adenylate at the end and cause a mRNA to be formed. And this occurs in transcriptional cascade, where the beginning proteins are transcribed more than the end proteins, so that they are in correct levels. This occurs in a primary step and a secondary step for the rabies virus. The primary step is the very first step, and it's very brief. And this is just to get the ball rolling until the secondary step, where there's more nucleocapsids. They accumulate, and they're used as templates so that more transcription can occur. And this occurs throughout the entire life cycle, secondary transcription. Now, with translation, I did mention that the RDRP is a completely competent RDRP, and that means it can transcribe and also cap the mRNA. Now, this is important because capped mRNA is required for translation at the ribosomes. Similar to a eukaryote, they form Cap1 structures, the viral RDRP, methylates the Cap0 structure into a Cap1 so that it can be able to be put into the ribosome and then translated as normal. Now, these are positive sense strand mRNAs because it is a negative strand genome, so it needs to transfer that to positive strand in order to be translated correctly. And the location of this translation occurs in the cytosol for proteins M, P, N, and L. And the G protein, which is the glycoprotein, actually goes to the endoplasmic reticulum, where it is then transported to the vesicles, and that will be expounded upon shortly.
Since the rabies virus is a negative stranded RNA, it needs to be synthesized into a positive strand. This action is done by using a complex from the N protein, which is a protein needed for replication, and the P protein, which is a protein that helps with solubility and proper folding. This helps nascent RNA to become encapsulated, which combined with RNA signals from the viral RNA polymerase causes sequences in the genome template to be ignored at each junction to create a full-length RNA strand known as the antigenome. The antigenome synthesis results in genome progeny of the viruses. Whether an antigenome or genome is used as the template, encapsulation is required for both. The antigenome also produces trailer RNA, which is a primary product in infected cells in the absence of new viral proteins. A secondary transcription occurs when the nucleocapsid accumulates in infected cells. This is initiated by a cis-acting RNA sequence, which governs the replication in the 3' end and serves as a promoter for synthesis. The complementary sequence is at the 5' end. The replication of genomes and antigenomes is symmetric for rabies. While secondary transcription is occurring, so is assembly of the rabies virions. The individual components are assembled in separate cellular compartments. The 3 important components of assembly are nucleocapsids, G protein, and M protein. The nucleocapsid is formed during RNA replication. The G protein is synthesized in the secretory pathway by ribosomes in the endoplasmic reticulum. The N-terminal signal targets the protein for insertion and is then cleaved. then transfers from the luminal side of the ER to form an ectodomain. After forming a G protein monomer and converting it to a trimer, it is transported to the Golgi apparatus via vesicles to the endocytic recycling component and fuses with the membrane. In the plasma membrane, the G protein is organized into clusters. The G protein is important for viral assembly and enhancing the budding process. Lastly, the M protein is synthesized as a soluble protein, which is then associated with the membrane. The M proteins of infected cells are organized into membrane microdomains. Then the cystotic and membrane-bound M proteins form the nuclear M protein complex. The virions are released when the M protein interacts with the host protein to create an MVB complex. Vesicles from the endosome membrane bud into the lumen and use a short sequence known as the PPEY, which is responsible for redirecting cell machinery to the plasma membrane. Another interesting thing to note is when a cell becomes infected, Negri bodies form in the host to create virus factories.
So with rabies being so deadly, and there's only 1 to 3 cases reported annually in the US, why would you want to study this virus? It's going to kill you. It's super rare. Why would you want to do it? Well, this is just looking at the US. In reality, 59,000 people per year die from rabies in 150 countries around the world. And this is actually an underreported number to be thought, which means it could be even larger than this. 95% of these cases also occur in Africa or Asia in places that are rural, low income, and are super susceptible. Rabies can actually be beneficial in the realm of therapeutics. And rabies has actually had, are thought to have been useful as vaccine vectors or cytolytic agents. One example being for HIV infected cells in terms of vaccine vectors, and then cytolytic agents for oncolytic viruses for cancer cells. Now, with studying rabies, there are hurdles that need to be jumped over, but even though there are hurdles, there are still breakthroughs that are being made, even recently. Some difficulties in rabies research is actually we're still learning a lot about rabies, particularly about attachment. In fact, recently there were 4 new variables added to rabies attachment, 2 of them being receptors, one of them being transferrin receptor protein 2020 that was discovered in 2023 to act upon rabies attachment. Same with metabotropic glutamate receptor subtype 2 in 2018. And then 2 mediators that are present in the cell membrane of cells, integrin B1, beta 1, and threonine kinase AP2, associated kinase 1 in 2020. Both of them were discovered then. They also mediate attachment. Additionally, like I had mentioned before, there's a lack of rabies education and accessibility of treatment. In a study in Cameroon, 11 people who were surveyed in the study who had been by a dog, only 2 of them sought care, which is really worrying considering how dangerous rabies is and how deadly. Despite these hurdles, there are some significant breakthroughs, particularly in the realm of monoclonal antibody treatments, because treatments for rabies are so expensive and so high risk that we want to make sure that we can find easier ways to do it. And that way would be a monoclonal antibody treatment. In a paper from 2022, it was noted that 2 neutralizing antibodies can stop G protein from forming a trimer that is what's needed to fuse out of the endosome at low pHs. Additionally, Uniform Services University also did a test on monoclonal antibodies grown in the lab that they called F11 that could potentially be used for a rabies cure. And these also work through neutralization. So in summary, rabies is a nootropic RNA virus that is spread through saliva and neural tissue. And with that, we have some remaining questions on how we can guide our research and how to better prepare and hopefully treat rabies in the future. So 1, how do we improve rabies education, possibly implement public health training for the general population? How do we create better access to treatments and medications for those people? What can we learn more about viral attachment, because that is a not particularly well-known aspect. That's the most important aspect to develop treatments. And then 4, will monoclonal antibodies be able to be used for rabies treatment in the future? We'll see. We hope so.
That concludes our presentation. Here are the references we use. Thank you.